السلام عليكم ورحمة الله وبركاته.
السلام ورحمة الله وبركاته.
مساء الخير جميعا. Before we begin, please confirm in the chat that the audio is clear. إذا الصوت كان واضح قبل ما نبدأ إن شاء الله. مساء الخير جميعا. وفي البداية أحب أبارك لكم الشهر الفضيل، ونسأل الله أن يتقبل منا ومنكم صالح الأعمال أعاده الله علينا وعليكم إن شاء الله بالخير واليمن والبركات. Good evening everyone. I'm here today with you. My name is Dr. Amani Gusti, consultant clinical biochemistry, and it's my pleasure to moderate today's session. واضح الصوت تمام. Welcome to this scientific webinar organized by the Saudi Society Clinical Chemistry. Actually, it's my pleasure or my privilege to announce regarding the first activity for this webinar and announce the official launch of the Lab Quality Accreditation Working Group established a few days ago this week under the leadership of the President of Saudi Society Clinical Chemistry, Prof. Anwar, and the STEAM Committee. Actually, this group was created to advance best practices in the laboratory quality systems, support sustainable accreditation, implementation, and harmonize quality practice across clinical laboratories in Saudi Arabia. So I would like to express my sincere appreciation to the President of Saudi Society and the STEAM Committee, Prof. Anwar Burai, for their continued leadership and support of quality and accreditation educational initiatives within the Society. We value the Society commitment to advancing laboratory excellence through structured scientific activities such as today's webinar. Actually, our webinar today is titled CAP Checklist 2025, All You Need to Know. Whether you are new CAP experience or already experienced, today's session is designed to strengthen foundational understanding of the 2029-25 CAP checklist across chemistry, toxicology, point of care, and all common. So this actually topic matters because CAP requirements are often approached primarily before inspections. However, checklist structure reflects core principles of risk management, quality assurance, and patient safety. This session actually aims to provide a clarity on structure, revisions, and compliance expectation in a practical and organized manner. So it's now my pleasure to introduce our distinguished speaker, Mr. Faris Jawatar. Mr. Faris is a supervisor of chemistry, immunology, toxicology, and point of care at the Ministry of National Guard Hospital in Dammam. He brings over 25 years experience in CAP accredited laboratories and has served as a member of the CAP Laboratory Accreditation Program Advisory Panel since 2014. Contributing to the annual review and auditing of multiple CAP checklists across major disciplines. Mashallah, he has received formal recognition from the CAP Council on accreditation for his contributions to several checklist editions. His expertise spans CAP, JCI, and ISO 15189 accreditation, preparation, quality control, implementation, method validation, and technical evaluation of all laboratory systems. Mr. Faris, we are pleased to begin your presentation. The floor is yours.
Thank you so much, Dr. Amani. I would like really to thank Prof. Anwar for invitations and Saudi Society of Clinical Chemistry. As you know, and everybody knows, chemistry is the mother and father of all sciences. Our body is chemistry. If any mess up with any of our chemistry, messengers, hormone, enzymes, then we have a problem with our body. That's why we are always proud to be a chemistry chemistry expertise. And really, that is a great effort by the SSC to start this kind of education sharing experience, and especially with the CAP checklist and CAP, and to bring awareness of the CAP checklist. And this really will make a meaningful for the journey of quality improvement and accreditation if we really understand well those checklists and be able to apply them the way designed by the CAP commissions and the panel on the annual review. Today, inshallah, we'll start with the 3 checklists, which is the chemistry, immunology, toxicology, and point of care. And then later on, continue with the other, the all commons and the general checklist. So it's very important, really, when we talk with the CAP checklist, to start with the basic, the foundation of CAP accreditation, which is the CAP inspection philosophy. And this really is, I found no excuse not to bring these 2 slides talking about the philosophy, because this is, I found really people need to be aware of what the expectation from us as inspectors, and the expectation from the inspectees, how the journey of inspection goes, in order to all benefit from it. As you can see here, the first thing about the philosophy is the peer-based approach. CAP would just really want us as a professional and pathologists, when conducting inspection, to treat our colleagues as peer, and then we come to your lab for peer review. This is very important. You have to highlight that, because really it will give a respectful atmosphere for us as inspectors, and we will inspect, and we melt down the pressure of the accreditation. And the most important from us as inspectors, because CAP required us to be a pioneer, to be expertise, sharing experience, to improve the processes. So when we go there, we really want the people to benefit from our experience, and we just let them, be part of what we have and what we carry on with us. And this really is very important as an inspection goes on. And the other thing is education. CAP really very concerned about the education, and that is the session should be really mutual between us inspector and inspectees. It should be interactive, and to sharing insights with the inspectees, and to identify if there any potential risk, and then helping laboratory to improve. And that is, the purpose of inspection. And also, as you know, quality is quality. Our work is all rely on quality, and we have to focus on the 3 phases of analysis: pre-analytical, analytical, and post-analytical. And that's what we do on a daily basis. We have to ensure safety, accuracy, and compliance with our standard and CAP checklist. Then we have continuous improvement. Of course, we audit, we work every day, we check what we do, we critique ourselves, then we find ways to improve. And inspection is not only for finding deficiency, but is the process to help the lab maintain high standard all time, every time. And then support, and the ultimate goal is supporting patient service and the outcomes of the daily work of us, which is make sure the patient safety is being met. And should be proactive approach of risk assessment to find out if any risk, and how to identify the risk before the failure, before the harm occurs. And this through the robust quality management system, and that is it will really encourage us to check on what we do on a daily basis, try to find, if any gap, we feel it ahead of time before, it occurs. And this is a continuous improvement, continuous checking. And this is the 5 really CAP philosophy, and we need to carry us with us every day, every time in our lab and when we go to inspect others. The checklist. What are those checklists? Those are the standards provided by the club and provide a clear roadmap and not only achieving accreditation, but running high standard quality laboratory. So if we follow those checklists, we are sure 100% we are really performing high quality laboratory. And that is the importance of understanding those checklists, those standards, and apply them on daily basis of our practice. Almost about 3,300 checklists. And the most important, those checklists are developed by about 500 pathologists and expertise. They have the commission, about 20 commissions, all the most skilled people in the USA and other parts of the world. And most of them are pathologists, specific for specific sections or disciplines. And then us as the international laboratory expertise, we do after the commissions, review those checklists, send it to us as international expertise. We go through every checklist and we have to send our input to the commissions, and then where the improvement come, the revision come, and deletion or adding new checklists. And as you know, there are 21 disciplines, chemistry, hematology, microbiology, anatomical pathology, and so on. There are 21 disciplines, and each discipline has its own checklist. For example, all commons, it has about 82 checklists. And the checklist, as we know, is organized by disciplines to make it easy to the staff and helping simplify the preparation for the inspection. And as you are aware, they are really detailed and rigorous, those checklists and standards. You need really to read them over time, and then over and over and over. And you have really even the notes. Every word in those checklists are considered part of the inspection. Don't really. Some people really rely on the title of the checklist and the compliance. No, as per CAB, every word in those checklists is considered part of the inspection. That's why you need to read the detail and indicating exactly what they require from the laboratory. Really, those checklists are very straightforward. They're telling you what we need from you and how to comply with the accreditation. And the laboratory require to have numerous, and you need to have policies. And those policies, it has to be covering all those checklists and then apply those policies in your practice. So you have the policy, and those policy has to cover those checklists, and then you practice those policies, and then you document them. Those are the three pillars of CAF inspection. You have policy and procedures, you practice them, and you document them. Then really you are in compliance with the CAF. And to ensure the quality of the laboratory testing and the patient safety. Those are the two main things all about the CAF accreditation. You have a quality of laboratory testing and quality result, precise result, accurate result, and then to maintain the patient safety all time. As you said, we have really 21 disciplines, and then we have universal checklist, which is the general checklist and all common checklist. The total, we have 23 checklists, and each checklist has its own, checklist inside. For example, chemistry, it has its own checklist. Hematology, it has its own checklist. And they are really written in a very in an easy language, and just need to be, take some time to understand between the lines. And also, they meant to remove any guessing. Whenever I go to inspection, I told people I don't want to hear, I think, or maybe, these are just checklists. Really, you need to be 100% sure. Either you know or you don't know. And if you don't know, this is our job as an inspector to explain to you. But there are really no guessing in the compliance of those checklists. And they are evidence-based and rigorous standards. Using detailed checklists, the CAB ensures that the laboratory practice align with the highest quality standards. Because CAB gave you the checklist, and then all you need to do to apply those checklists into your daily practice and to document them. As simple as that. We don't need you to make any guessing or go beyond the checklist. If you go beyond this checklist, that's fine. We accept it. But really, we need you really, whatever written in the checklist, it should be practiced and should be documented. And they are frequently and they're sometimes exceeding the regulatory requirement. If you have national regulatory, and those checklists really sometimes they're exceeding the expectation of your national regulatory. The customized, as you know, we have a customized checklist and we have a master checklist. Master, it has everything customized what you do in your laboratory. So prior to your inspection, you will have your customized checklist, cover what you do in your laboratory. And then an inspection, you will be inspected, you know, there is no surprise. CAB checklist send you the cover your lab menu, laboratory testing, and then the inspector will have the same checklist. So there is no surprise, and the staff will be prepared for the accreditation, and then based on the requirement in the checklist, and serial applied uniquely to your laboratory. And checklist is compatible with today's documentation control system, and that's the thing. This is one of the common also deficiency to have a laboratory control system. So when I go to your laboratory, everything you do, it has to has traceable to your policy and procedure. Every form, every thing you do, it has to have, you know, a quality control system that I would know this practice belongs to which checklist. So it's very important to control your practice through a quality control system, and it will be traceable at any minute, at any time. So I don't need to look. So that's very important to have this quality control system for your policy and procedures and documentation and your practice. And as you know, it will come as Excel, Word, and PDF. So I brought this just as a reminder because really this come in few years, this logo of policy and procedure. This icon always represent policy and procedure. Now I brought in two checklists. One checklist is for calibration procedures, and the other checklist is calibration verification materials. The difference between those two, the first one, it must have a policy and procedures, either a single or within other policy and procedure, but this has to have policy and procedure, calibration procedure, and then it will tell me when you calibrate, how often you calibrate, or what kind of calibrators. And the other one is just calibration verification materials, and this can be showing what kind of material you use. It can be get it from the insert sheet from your calibration box. But this one, it must have some procedure in details about the calibration procedure. That's just, I brought it just a reminder for our staff, maybe some people new to the CAB. When you find this logo, you always make sure you have a policy and procedure, cover this checklist. This is Dr. Bruce Williams. What he says, this is our lab 31st president. He said the CAB equipped with knowledge, skills, and resources to remain current and relevant in an evolving healthcare landscape. So those are really a very profound, word, knowledge, skills, and resources. And that is, our role and our responsibility as expertise CAB inspector. When you go there, really we have to share the knowledge, and we have to apply our skills in the inspection and help the people to live their skills to the level of the requirement by the CAB. And then the CAB, they have all the resources to remain for the laboratories. So whenever you send email to the CAB, they immediately respond to you. If you send to us as an inspector, we respond to you. So, and really the most important is the evolving healthcare landscape. And as we know, the healthcare is developing day by day, and that's why CAB is always prepared to match with that and for the sake of the patient safety. Now we delve into our checklist. So the first, we have a new checklist, CAM. Uh, 12, 9, 25. This is for hemoglobin A1C. We need to do.
مستر فارس الصوت the voice صوته بدأ يقطع عندك مستر فارس. Mr. Faris?
Aywa.
Are you okay now?
Yeah.
Okay. So it's clear?
No, it's clear. Everybody.
Yeah. Okay. Yeah. Okay. Yeah, it's got it. Okay.
Yes, and the CAP says for this one, the accuracy of hemoglobin A1C result due to limitation of the product stability, this may not be available outside the United States. Okay, and then there is a checklist or the accuracy-based material. It made by the Center of Medicare Medicaid Center CMS have established acceptable criteria, which is 8%. In this checklist, what it's telling us, there are two acceptable criteria or performance acceptance criteria, which is 8% and 6%, which means we're going to have two CAB PTs, one with 8% for evaluation criteria by the CAB, or when you have unacceptable result, which means 6% means with accuracy 94, and this one with accuracy 92. And CAB says the 8% is only available for the Canada, United States, because it has, stability problem. That's why we know that if any material, it has a stability, of course, it will affect the accuracy. That's why the accuracy here is 92%. And when it is stable, of course, the accuracy will be better, which is 94%. And CAP says here, the record of accuracy-based evaluation. So this plus minus 6%, it is for international user, and plus minus 8% is for the United States. That's why I went here to the catalog and get this to checklist. Here's the definition of what is the accuracy program. The CAB uses the accuracy-based program to verify the accuracy of your test result. Again, is a gold standard. So the gold standard here is set by the CMS, which is the 8% acceptance criteria, and CAB is set 6% acceptance criteria. The accuracy-based program uses challenges specimens that are matrix-related, bias-free, and have target value traceable to certified. The same matrix, the word matrix, it means always use the same material. For example, if the serum, that means the the gold standard or the material. And then here CAP, they give us 2 checklists. Can you hear me, guys?
Yeah, now it's okay.
I think the problem from me or from you guys? There is
no, net, yeah, disruption.
Okay, so all right. The CAP here, they give us 2 checklists. The checklist is 51, this for international, with target 6 plus minus 6%, which means if you this is going to be your evaluation criteria. This is when CAP says sample, they have the mean of all the peer group plus minus 6. It will give you the acceptance criteria, and this for people outside the international. So for us, we order this one GH51. But for the people in the Canada, United States, they have 5, which is it has 8% evaluation criteria. This will be evaluated your result with the CAP peer group, or this for the United States and Canada. And for us, it's 6%. Even if we have result unacceptable, we compare with other lab, we use plus minus 6% as criteria of evaluation. And this one of the checklists, we need to make sure we understand that. We understand that there is 2 criteria of evaluation, which is 6% and 8%. So we will not be confused to order a wrong one. Then we'll have sample problem stability. It will give us, unnecessary, unacceptable result, and then lead into unnecessary troubleshooting. This is the first checklist. The second checklist, it is CAM 15225. It should talk about the AGFR and LDL calculation. It says here the clinician have access to information regarding the equation used to calculate result for estimated AGFR and also for LDL. It says here, a note says, calculated result may differ based on which equation you are used. Either you use the equation of MDRD or use the equation of CDC, CKD, ABI. Either way, you need to make sure that the physician, they have access to the equation you used and what kind of equation you use. And that's a requirement by the CAP. And this could be either through the laboratory information system. If you have a laboratory information system, you need to include this equation with the patient result. Or if you don't have a laboratory information system, then you have to send a memo to the physician and telling them for the calculation of AGFR or LDL, we use this equation to be reference for the physician. And that's a requirement by the CAP. Patient report with information on the calculation, it has to be there. As you said, either electronically or by memo, it should be with the patient result. Test reference guide or other mechanism to providing calculation information. So CAP said either, but at the end, the physician or the clinician, they must be aware on what equation you used and the calculation of the LDL and AGFR. We have here revised the checklist for the toxicology for opiates regarding the cutoff. The urine opiates immunoassay cutoff is appropriate for clinical setting. Here they're talking about the clinical setting because, as you know, toxicology, you have clinical and non-clinical. Now, opiates class immunoassay are primarily designed to detect naturally occurring opiates, which is morphine and codeine, and have varying cross-reactivity to the semi-synthetic opioids. Therefore, when utilizing for clinical care, including support for emergency and pain, we have to use a cutoff of 300 nanogram per ml cutoff. So CAP says here for immunoassay, for the opiates, for the patient and healthcare, you have to use 300. For non-patient, non-clinical, you have to use the 2000 nanogram per ml. This is the revision of the checklist. And then here the notes, it's recommended that the laboratory review the package insert for the OBS immunoassay for cross-reactivity with the service. I think we have to go to the basic, which is the insert from the manufacturer. And as you know, CAP says always follow the manufacturer instructions. So a specific immunoassay for detection of semi-synthetic and synthetic, it should be always for the drug required, or if you have alternative target method or like mass spec. So the CAP says here for the evidence, patient report with an opiate immunoassay cutoff for the clinical setting. And we are going to use the cutoff 300 for patient care, and it has to come with the patient result. So CAP says for patient result, you need to include the cutoff for opiate, which is 300. That's the compliance of this one. While in the previous one, this is the previous one, CAP says compliance written procedure. Here just asking for the written procedure, but for the new revision, it's a must to have the cutoff value with the patient result. That's the revision from this one. But in the previous one, only written in the policy and procedure, so it doesn't need to be with the patient result. This for the, we finished from the, that's the, we have the, for the chemistry, then we have the POCT checklist. So we have POCT for competence assessment for wave test. As you know, we have waved and non-wave tests. As you know, the definition of wave, the simple procedure. It doesn't need trained people. It can be by run by bedside. Doesn't have to be run by the laboratory people, and it doesn't need maintenance. Those are the small devices like glucometers and uresis for the urine. And now we have more for the INR and creatinine. Those are the wave tests. And also, although CAP here says the competency of personnel performing wave tests is assessed on each. So when we have the wave tests applying in the healthcare, CAP require some competency evaluate individual ongoing ability to knowledge and skill achieve the 10th. Competency must be assessed as the following by the frequency. It can be assessed annually and thereafter, or it can be performed throughout the entire year to minimize. So it's up to you in the laboratory. Either you want to perform it annually or you want to perform it throughout the year. When the problem are identified with, if you have a problem with staff performance, you can also do competency. If more stringent, if required by the national accreditation agency like SBAHI, you must be following. Also, CAP, you said you have to follow your national accreditation standard. For wave test system, the laboratory may select some of which elements assess. Yani, here for wave test, CAP says it doesn't necessary to go through the 6 elements. We will discuss in the next slide. And you can use 1 or 2, 3. It's up to you. Yani, it's left it up to you to select which one. But for non-wave, it's a must to apply all 6 elements. These are the 6 elements for the wave and non-wave test. The first one is direct observation of routine patient test performance. And you have to have, always we ask examples. You can pick up any patient result as example with the patient identification and patient collection if you are working in the receiving, or in the lab processing and identification, like pre-analytical identification. Choose one of the samples, put it as example, and it has to come with the staff ID. If you have electronic system, print it or keep it software with the staff ID. If not, then you have to print it out and let the staff put ID, because competence applicable to every single staff. Monitoring, recording, reporting system for the result. Also, you have to show any result, especially critical result with reporting with the staff ID. Also, review intermediate result worksheet like control PT. Also, you have to have a control record PT record or maintenance with the staff ID as well. For the number 3, number 4, direct observation performance for the maintenance function check. Also, you need to have also example assessment of the performance through testing previous analyzed specimen, internal blind sample, just like CAP samples. Also, you have to have the staff ID. And if you have any problem with the instrument or any OVA, ECR troubleshooting, you need to do it on staff ID. Those are the 6 elements of the competence assessment, and this is required for the non-wave and wave test. For wave test, CAP will give you exception and leave it up to you either to apply 2 or 3 or whatever you based on your policy. But for the non-wave test, you have to apply them. What we require from this record of competence assessment for new and existing testing personnel reflecting the specific skills assessment and method evaluation. That's what we do in our POCT when we have a new staff, especially the nursing, they are the one using the POCT, we have the competency assessment for them. And that's the compliance with this checklist. This is the current checklist, okay? And as you can see, the difference between this one and the other one here. There is record of competency assessment for new and existing testing personnel, reflecting the skills, assessment, and the method of evaluation at the required frequency. You have here to mention the frequency. While the previous one, there is no frequency. That's, the difference between both of them, and this is the revision. This is the current one. The CAP added evaluation at the frequent. They have to specify how you do evaluation, every 6 months, every annually, but here they left it just open. And that's why, the difference between 2 checklists, the current one and the previous one. Here, CAP required, specific, the frequency, it has to be mentioned in your policy and in the practice. This current checklist, we have another also for non-waived, the same checklist, the same 6 elements of the competency. But here, the difference in the current one, the CAP says this assessment may be done virtually if allowed by national, state, or provincial and local laws and regulation. Here, what the change CAP made, this actually come into effect after Corona, and some people, they might do it, inspectors, they might do it virtually to check for the record of the competency assessment. And now CAP allowed. This is the sentence they added into the competency assessment. So the record, again, it needs also frequency, but allow you can do it, virtually instead of going into the lab on site. That's the difference between the current and previous checklist. And this is the previous checklist. As I said, it doesn't have this sentence, which is the same. Everything the same, except this adding new. They allow you to check the competence assessment. You can do it virtually. So we have a new checklist for the POCT, specimen handling of whole blood based for coagulation. As you know now, coagulation really, it becomes into effect into the point of care, which is really helping a lot. They say the sample based on coagulation testing be handled according to the manufacturer. As you always CAP, for POCT and other everything, you have to follow the manufacturer. The specimen must be not heated or refrigerated or frozen, should not centrifuge. The specimen must be rejected because using whole blood for additional specimen handling for platelets function. So this is a new checklist for the coagulation, and this is the stipulation by the CAP. You need to follow it. We have another coagulation testing for therapeutic anticoagulant recommendations. The recommendations are available for clinician. Here, CAB required you have to communicate with the physician, and this information should be with the physician. Either you say as a disclaimer with the patient's result, or through the communication through electronic memo, or if you don't have electronic, then you have to send a memo. So it's very important to share this information, especially with vasoelastic testing, therapeutic range, and this all should be available to the physician. The coagulation tests available to physicians should be applicable to the anticoagulant drugs in use, and information is available on the test values that indicate that the anticoagulant is present and the therapeutic range are when available. So this should be very important. Every result with the therapeutic range and those information should be available to the physician. When I come to inspection, I want to make sure those information are shared with the physician. Either you share it to me with the patient result, or electronic communication with the physician, or with the memo with the physician. But you must show me those information the physician are aware of. And this is continuation of the previous one. For the vasoelastic, if you have monitoring antiplatelets or anticoagulant medication, then you have to have a vasoelastic testing recommendation on the utility of testing in clinician meaningful situation must be available. Again, all this information should be available to the physician. So the compliance of this one, either, as I said, either through memoranda to the physician, test reference guide, interpretive comments on patient report, or other mechanism of recommendation. So the bottom line is this information must be shared to the physician. And as an inspector, when I came, I need to prove to me all this information are physician aware of and share to the physician. And you have to show me evidence, either electronic or paperwork. This is at the end of the. I hope that I'm compliant with the timing. This is the end of the session today, and still we have some work to do with all common checklists. It has lots of revision and new and the general. This might be in the upcoming webinars. And thank you so much. And we'll open the floor to Dr. Amani.
Yeah, mashallah. Thank you, Mr. Faris, for this clear and insightful overview. Actually يعني كثير ما يتعامل مع الكاب تشيك ليست just on time يعني عند اقتراب موعد الفيزيتنج او الزياره التفتيشيه. But actually هي في الواقع يعني تعتبر operational quality tools او ادوات تشغيليه تحدد معايير الجوده والكومبلاينس الامتثال داخل المختبر. فكان هدفنا اليوم من هذا الويبينار تبسيط الصوره يعني وبناء فهم منظم لهيكل التشيك ليست والتحديثات الحديثة. So we will now move into questions and address. MashaAllah, we have so many questions here and just selected the questions. To begin, first question, from your experience, what is the most common challenge laboratories face when reviewing updated CAP checklists? Mr. Faris? Mr. Faris, are you hear me?
Yeah. Hello?
Yes.
And I hear you, but you don't...
question again, from your experience, what is the most common challenge?
Can you hear me?
Yes, I heard you. Maybe network عندك shall I repeat the question?
Yes.
Mr. Faris? يبدو أن النت عنده في مشكلة. أوكي. طيب. Stop sharing. Yeah, stop sharing the presentation. Stop sharing the presentation, Mr. Faris.
Hello?
Okay, yeah. Are you hearing me now?
Yes, I hear you. Yeah, I got the question. Yeah, the most issues with when we get a new or revised checklist, honestly, and that's the common thing we experience during our inspection. People, honestly, they don't spare more time reviewing and discussing those checklists and understanding the content of the checklist. Really, you need to go through the contents, just like the first one I shared with you. It has two acceptance or performance criteria, which is 6% and 8%. If you order the wrong one, then you will be having into unacceptable, running into spending some time, troubleshooting, unnecessary time waiting. So the most important, you need to read and then ask. If you don't know, ask. There are people, lots of people in this country, really expertise and knowledge. And the issue is the CAB is the understanding of the checklist, because really institutions are not, investing into educating people about the checklist. They're investing into getting the PTs. But really, I found out really people not spending time, try to sit down, reading, reviewing, and understand the checklist. Yeah, that the thing needs to be addressed to the people.
Okay, more question regarding first for cut-off verification. Do we need to verify cut-off verification? Do we need to verify positive, negative, intermediate? And how many sample we need? How many sample we need?
Yes, this is also one of the challenging. So let's go to the basic. What the CAP basic is very clear, very straightforward. CAP says follow the manufacturer instruction. The manufacturer said if you look into any insert, they said you need, even CAP says in the checklist, you have to have two value. One above the cutoff is verify the positive value, because the cutoff is a qualitative result based on the quantitative value, which is the cutoff value. And then you have to another value less than the cutoff, which is verified the positive. Then there is a gray zone, is not in the CAP checklist, is not in the manufacturer insert. Some people, they say 20% above and below the checklist. So above, I'm not worried about it, because if you have positive, that means it will cover the gray zone. But some people, they said concern below the cutoff 20%. And honestly, we have one of the new instruments in our lab. We are trying to get the gray zone below the cutoff. Honestly, maybe how many years we never get a gray zone. And then this is, as I said, you're exceeding the cap by creating some policy, a gray zone. Then you need to verify. You put yourself into verification, which is non-exist. And yes, when I come and ask, even in our lab, we couldn't find. But you can have, what we call it addendum report. We're saying that our lab applying 20% below and above, but we don't have samples, so we satisfy with the report of above and below the cutoff.
Yeah, perfect. Okay, also here I have a question. They need explanation a little bit about annual review process, when it started, how it works, and what your role as reviewer is.
Annual review.
Yeah.
If I get more.
When it started, how it works.
Competency?
Yeah.
You mean the competency? Yeah, the competency is stick with the basic. You have the 6 elements well explained by the CAP. Get those 6 elements, put them in the form, and then apply them one by one with the documentation. And CAP says you can do it at the end of the year or through the years. CAP says in 12 months you have to have competency assessment, or if you have problem. But the easiest way, we have it in our lab. We build the 6 elements in form with process control, and then one by one is documentation, as accession with the QC and the this is the easiest way to be compliant with.
Yeah, perfect. Also, question, you mentioned in the hemoglobin A1C revised checklist with 2 BT.
Yes.
Each has its own evaluation criteria. And what happen if an international ordered the BT with 8% instead 6%? How CAB would evaluate?
Yes, this could happen. It's possibly happen. Now you put yourself in trouble, okay? Because this 8%, you know, the accuracy is only 92%. It's the sample. It has a problem with the stability. You might end up an unacceptable result. Then you need to go into BTCN. You need to go into acceptable criteria. But what I will do in this case, if I failed, I will do troubleshooting, and then I write comment to the CAB. We just order by mistake a wrong one, and then we ask the CAB to replace. And CAB really the cooperative in this one. Yeah.
Okay. One more. Yeah. Okay. Thank you. One more question for A1C also. How many samples we need to calculate the cutoff of hemoglobin A1C?
The cutoff. Hemoglobin A1C is not based on the cutoff. Cutoff always is the quantitative result to report the qualitative. Hemoglobin A1C is based on the HPLC, which is the column, which is the ions measuring the concentration. It's not really, there is no cutoff for A1C. It's just the direct measurement of the result.
Okay, and there is a question also for point of care. Point of care, how to document the monitored result of point of care?
Yeah, this is one of the challenging, because the point of care is very challenging. It's done with in the other side with the nursing. That's why the role of the quality control or the coordinator of POCT, the regular really monitoring. I always recommend our coordinator to go on the regular basis, check, make sure the results are correct, the maintenance is done, and if any issue should be addressed immediately. So it needs really regular monitoring, because when it comes to the other practitioners, they are not lab, really are not oriented as a lab with our documentation, rigorous documentation. We need to be really visiting more often and make sure everything under control with the documentation.
Perfect. We will take a question for chemistry. Is it mandatory to use the calculated LDL, or direct LDL can be enough?
No, it's up to you. CAB, as you see in the CAB checklist, you have both in the reporting. If you can use a measure LDL, but some labs really using calculate LDL, then it have also PT reporting results. So it's up to you which direction to do. But if you use LDL, CAB says in the new checklist, you must document the equation you use with the patient result.
Okay, questions. What is the most frequent misunderstanding laboratories have about updated CAB requirements?
Yeah. As I said from the beginning, the updated CAB requirement really need to be addressed immediately. This is a role of the laboratory QI officer. Really, it has to be immediately to be addressed. It has to be immediately to be discussed within the lab, and then to get to the what's updates and what the compliance needs to be implemented, and what the document need to be updated. So any update from the checklist, it has to be updated in the document. It has to be on the updated the practice. It has to have awareness with the staff, and then the documentation. So is a teamwork has to be done immediately upon receiving of those checklists.
Okay, and there is also one question for verification process. What is the verification process for latex test? For example, ASOT latex.
Okay, the latex test at the end, the reporting is a positive, negative. The question of the verification, is it the verification with the validation or verification with the CAB result, is not clear. But if we go with either, because this is a qualitative result, To verify the result, it means either, if you mean by the new test comparison, or with the CAB result with the peer group, the question is not clear. Either talk about the validation, yes, you have to have a positive and negative result. This should be given positive, and it should be given negative, just like, qualitative comparison. And then, or if you talk with the CAB, then your verification through the peer group. If the peer group negative, should be negative. If positive, should be positive.
Okay. Can you please just give us a little bit about body fluid checklist?
Yeah, this is one of the challenging, and we have really rigorous discussion every year. I really very, adamant about this body fluid. I always speaking to CAB, all the experts, I said, guys, we know that for body fluid, in order to get a normal sample, you have to have intervention. And this intervention procedure, nobody will volunteer. So what we do, we collected this sample from the patients. And basically, is not what CAB meant to do, especially for the reference range. Yes, we can do for the comparison, that's easy. You can get body fluid from microcollect, whatever you have, 5, 6, it doesn't matter. Do comparison, that's fine. And for the body fluid, we use the same reagent as the serum. So you cover for the AMR and we cover for the other thing. But the most challenging is the reference range, and we use the disclaimer, and CAB even add for the disclaimer the validation. You can use the disclaimer by CAB, there is no reference range and there is no validation. This should be with the patient result if you have electronic system. If you don't have electronic system, you have to send the memo with the physician, informing them body fluid is not being validated, no method verification validation, and there is no reference range. Therefore, you have to compare the result with the serum. So, but you do what you can. But really, when I go to inspection, I don't really go deep into this one, but I ask what you do, because even CAB do this.
Okay. Does calculated test should be included in proficiency testing like LDL and EGFR?
Yes. And there is PT for that. There is PT for EGFR and PT for LDL. Yes, there is calculated and there is, direct measure. Yes, it's there, and you have to have. CAB says every practice you do, you must have PT. If CAB not cover PT, then you have to do alternative testing. But this is one of the common deficiencies, that the variation between your lab test menu and CAB activity menu. So some people do some practice is not in CAB or vice versa. So you have to match 100% what you do should be reported to CAB, and to match with the menu, your lab menu with the CAB activity menu. But everything must be covered by CAB. If no CAB BT, then you have to do alternative assessment with other laboratory.
Yeah, actually we have maybe basic question, and actually we need to answer it for any basic. What kind of a proficiency test that we need to be performed and shown to the surveyor?
Okay. For the surveyor, actually, this is one of the things we always advise our colleagues. We always go with the book. Whatever customized checklist we got by the CAB, that's one we apply in the inspection. We don't need to carry our own practice, but we recommend. So everything we do, as I said, it has to have a CAB PT cover it. If you don't have CAB PT, then you have to have alternative assessment. But it is a responsibility of the inspector. Really, they have to make sure, and they have to be not go too hard on the inspectees, and don't ask men more than what they expect. It's very simple. Whatever you have done, it have to have CAB-PT. If you don't have, then you need to have an alternative assessment. And then we have to go with the checklist. We don't need to exceed the checklist to beat the lab into a dilemma of they don't know what to do. We don't need to confuse them. Just to make it easy and simple.
Okay, more question. Do we need to do all the wave and non-wave list?
Yeah, based on what you have. The wave, it has maybe 32 or 30 tests. Well, we have creatinine for creatinine wave, the INR wave, beta HCG, urine glucose. Whatever you need to do, you have to do it. But you have to select. And you cannot apply all of them. But now really the point of care or the wave that are really become very practical, and there is improvement in the methodology, and really most of the institution rely on them. So the more, but the thing is you have to do a regular verification and compare with the main lab to make sure really what we do in the wave test is really matched with your practice and keep under control and continuous monitoring. But you can apply as much as you can in your practice.
Okay, maybe now we can take last two questions. Actually, we have so many questions. We hope to cover all of them. Do you have a personal impact updating, revising any checklist? Some detail of any?
Yes. Actually, I've been involved with CAP since 2014. I involved in 5 checklists. I have personal impact on them. So the first one is for the volumetric glass pipettes. And this is one of my visit to one of the laboratory. I went for the just preparing for mock inspection. In the previous checklist, CAP says if you have to use glass pipette type A for it. CAP says must be used for preparation of calibration, preparation of QC, preparation of PT. And the CAP used the word must. When I went to that lab, they used Eppendorf. And we went there I said, why using Eppendorf? And they said, we have certificate, the share certificate. Then I brought the checklist. The checklist said, must use type A glass volumetric pipette. So I told them, this is really a very clear citation, but I recommend you write to CAP. Maybe CAP would allow that. And then those lab, they sent to CAP, they said, we are using Eppendorf, while the checklist said glass A. And CAP says you can use Eppendorf as long as you have the certificate and calibration. Then during the annual revision on the comment, I wrote to the CAP commission and with our peer group and the expertise about 67 people. I said, guys, CAP says in the checklist, strong language must, when you need to prepare for a QC, calibrator, PT, you must use glass pipette type A. But there is a recommendation from CAP. They said you allow for the Eppendorf. I said, so we have 2 options. I said, either we stick with the current checklist, and then we must use a glass pipette. If anybody use other than that, should be cited. Or we should remove this strong language and then keep it open. You can use any Eppendorf or any volumetric or any measuring devices. And then CAP, they change it really, and they remove the must, and they remove the calibrator and control, and they left it open. But they said you need to have the certificate of the pipette you use for the preparation. This one of the change I made for that one, and the other one also for the new lot for the reagent new shipment. The CAP previously said new reagent lot, and I told them we have to add a shipment because it can be the same lot come with the shipment, but it comes with shipment, and then they.
This is the most common issue actually before inspection.
Yes, and then also I convinced to exclude the cholesterol from the reference range verification. I saw the cholesterol is a common and is really depend on the population. And I told them 90% of the population their result above the reference age. You cannot, and then they agree. They said they really depend on the population, and then they exclude the cholesterol from the reference. They said you can use the manufacturer, and then the cutoff also. And I added the word quantitative. I told them it is a qualitative, but it's based on the quantitative value. So the CAP, that's why I adopted they said for qualitative tests that use the quantitative. That's one of the things also I contribute with the CAP. And then the last one is when you evaluate the unacceptable result. CAP says in the previous one, you need to do alternative assessment with split samples, with in-house method. And also they said recommend to investigate the bias, recommended. One of the, if you notice in the inspection, some people, inspector, cited laboratory for the bias. Then when in the annual I said, for the CAP, you said recommend. And it's being used for the citation. And then CAP fixed that. And then they said it's recommended but not required the laboratory to investigate the bias. So they make it open, and that's make it easier because you already passed the PT, but you have a bias above and below. So this is one of the contribution I made, which is been accepted. I'm glad that has been done.
Okay, one more challenge question with the blood gases. If there is no comparative CAP accredited lab in region to do comparison analysis and validity of patient sample, is also challenging?
Yeah, it is, because blood gas. As soon as you open it, it is, and the factor of blood gas, you want to avoid the bubbles and the timing for the sample. You don't want a sample to be clot. So it's very challenging, but what we recommend, you always ask the manufacturer to get you verification materials. Then you can have double, or you can have double PT. Then you run your sample here and send it to the other lab and make comparison. But one sample is very challenging, is very difficult. Always have duplicate sample for comparison and verification.
Perfect. What can we do if we don't have another machine to make the semi-annual comparison with it in our region? I'm selecting basic, intermediate, and advanced question.
Yeah, the semi-annual usually is only required when you have two instruments. The comparison between two instruments, if you have, but if you have one, you don't need to do it. It's only required if you have two instruments, either the same company or different company reporting the same result. And as a CAP, we always recommend to have a backup instrument. And this is one of the also challenging questions. When you go to the laboratory, they have one analyzer. They said, if this analyzer is out, what's the contingency plan? And that is but for one instrument, you don't need to do a semi-annual comparison.
Perfect. Thank you, Mr. Faris. Actually, we have many questions. So if you mind, share your email. They're asking also for your contact mail. They can ask you there. And it's actually excellent comprehensive overview of the 2025 CAP checklist update and compliance expectation. Today's sessions provided an important foundation for understanding how checklist structure, revisions, and operational requirement align with daily laboratory practice. So tonight, actually, we officially launched the Lab Quality Accreditation Working Group, and it's actually initiated the CAP Mystery Series 2026 by this year, because we have a question here. They need more sessions, more presentation, more webinars. And this marks the beginning of a structured effort to elevate lab quality, accreditation, understand, and quality harmonization. So this webinar marked the first activity for the Lab Quality Working Group and the beginning of the CAP Mystery. As I mentioned now, we look forward to welcoming you to the upcoming sessions, inshallah. Attendance certificate will be issued to all participants within 24 hours. And thank you all. Thank you, Mr. Faris. Thank you to the Saudi Society Clinical Chemistry and the STEAM Committee for your engagement and participation, everyone. Wishing you a pleasant evening. Thank you, everyone.
Thank you so much, and thank you for the effort put forward by Professor Dr. Anwar and you. And I hope this continuation really continue to share the knowledge and skills and experience with our colleagues all over in Kingdom and outside the Kingdom. Thank you.